Devin,
Good
shall we start?
Yes, Madam Visha.
Do you
Good
do
af
you think, yeah,
Okay, Madam.
all the presenters got the correct this address, this Zoom link?
Yes, Madam. We sent it to the chat.
But the chat, I don't think that everybody was there at that time, no?
Yes, Madam. I'll check it with that. Sorry, Madam. Give me a minute.
Yeah.
Madam, most of them are here, and we have sent emails to the other participants as well. So, Madam, we'll be starting the session
Yeah.
now.
Yes,
So,
please.
good afternoon, everyone. With great pleasure, we welcome all of you to the second part of the biodiversity conservation and management session. First of all, I would like to welcome our chairperson for the session, Professor W.A.D. Mahaulpata, Head of the Department of Zoology, University of Sri Jayewardenepura. Professor W.A.D. Mahaulpata specialized research interests are wildlife management, ornithology, wildlife ecology, wildlife conservation, and population dynamics. So without further ado, I would like to invite Professor W.A.D. Mahaulpata to take over the session. Over to you, Madam.
Thank you very much, David. And I would like to welcome you all on the behalf of the organizing committee of the Forestry and Environmental Science Symposium 2022. And I would like to thank you all because there are around 5 presenters. There are 5 presenters for this session. And since we are behind the time now, we will just invite the first presenter, so Mrs. Abe Siri, to give her presentation. I think David will share the presentation with us. Devin, are you going to share or the presenter?
Madam, our host will share it, Madam.
Yeah, please. Thank you.
Thank you.
Cytotoxic effects of microcystin LR, microcystin RR, nodularin, and cylindrous spermopsin on V89 kidney cell line. I'm H.S.N. Abesiri, Center for Water Quality and Algae Research, University of Sri Jayewardenepura. This is the outline of the presentation. Moving forward to the introduction. Cyanobacteria. Cyanobacteria are the most primitive prokaryotes that contain chlorophyll A. They evolved around 2 billion years ago, and research began in the 19th century. Around 2,000 species from 150 genera have been reported, with 40 of them being toxigenic. Some species form blooms. Blooming is confronted with a global issue. Contamination of natural water bodies by cyanotoxins is a worldwide problem, causing serious social, economic health problems and aesthetic nuisance, depletion of dissolved oxygen in deeper layers, and unsafe drinking water. It is not only a health hazard to humans, but it is also a health hazard to livestock. Cyanobacteria toxins can be classified according to their mode of action: hepatotoxin, neurotoxin, nephrotoxins, and skin irritants. The commonest cyanotoxins are hepatotoxic microcystins, nodularins, and cylindrospermopsin. These are the general structures of microcystin, cylindrospermopsin, and nodularin. These cyanotoxins can be accumulated in fish, aquatic plants, rice, and water. Therefore, human can be exposed to cyanotoxins by way of consuming contaminated fish, aquatic plants, rice, and water. In Sri Lankan scenario, there are limited studies regarding cyanotoxins. Because of that, in vitro studies are extremely rare. These are the world outbreaks due to cyanotoxin contamination. Therefore, WHO established a guideline for microcystin LR in drinking water as 1 microgram per liter and cylindrospermopsin in drinking water as 2 microgram per liter. There are no studies, no guideline values for nodularin. It is regarded as a microcystin value. In 2013, MCLR and cylindrospermopsin were included in Sri Lankan drinking water quality guidelines, with similar values included in WHO guidelines. Significance of the study. Microcystin LR and cylindrospermopsin are one of the most widespread cyanotoxins in freshwater bodies of Sri Lanka. It can be accumulated through the consumption of contaminated drinking water. Therefore, it can be created health problems to humans and animals. In our knowledge, limited studies have been recorded in relation to in vitro toxicity studies of microcystin LR, microcystin RR, cylindrospermopsin, and nodularin in Sri Lanka. Therefore, in vitro toxicity studies were carried out to find out the cytotoxicity effects of microcystin LR, microcystin RR, cylindrospermopsin, and nodularin in Viro 89 kidney cell line. This is the objective of the study, to evaluate the cytotoxicity of microcystin LR, microcystin RR, nodularin, and cylindrospermopsin on Viro 89 kidney cell line. Moving forward to the materials and methods. The following is a schematic diagram of preparation of Viro cell culture frozen from frozen stocks. This is the method of passaging of Viro cells. The MTTS schematic diagram can be seen here. Finally, the respective IC50 values were calculated. Moving forward to the most important part, results and discussion. The highest cell mortality percentage was recorded for cylindrospermopsin exposure to 0.5, 1, 5, 10, 50, 100, and 200 micromolar concentrations. Descending order of cell mortality was found for nodularin, microcystin LR, cylindrospermopsin, and microcystin RR at 200 micromolar concentration. Significant dose-dependent cytotoxicity was found. The results of the study further revealed that the cylindrospermopsin had the lowest IC50 value, 98.5, and the nodularin had the highest IC50 value, 160.1. Therefore, cylindrospermopsin was most toxic. Conclusion. According to the findings of this study, cyanotoxins have cytotoxic effects on renal cells. Among the tested cyanotoxins, cylindrospermopsin was most toxic to renal cells, while nodularin was the least toxic. These are my references. I would like to acknowledge National Research Council for the financial support and Center for Water Quality and Algae Research, University of Sri Jayawardhanapura. Thank you, all of you.
Yes, thank you very much, Mrs. Abesiri. Are you available with us?
Yes, madam.
Can you on your video, please? Yes, Mrs. Abesiri was so interested study, and I have some clarifications on that what you have done. Why did you select these cyanobacteria, these microcystins and these nodularin and this CYN and those? Why did you select particularly these?
Yes, madam. According to literature, these cyanobacteria were affected to the kidney renal failure. Therefore, we want to determine cytotoxicity effects for the kidney cell line. Therefore, we selected these cyanotoxins.
Actually, you have selected nephrotoxins, isn't it?
Yes, madam. Yes.
From these, and that you want to do this in the cell lines and checking.
Yes, madam.
So it is the novelty of your research?
Yes, madam.
And how can you explain this, the difference between this CYN and this NOD's toxicity?
Yes, madam. CYN is there are WHO recommended limits for guidelines limits for the CYN and MCLR. For MCLR, 1 microgram per liter in drinking water. For cylindrospermopsin, CYN 2 microgram per liter. But according to WHO guidelines, they didn't do the guidelines value for NOD. They recommended this value is same for the MCLR value.
Okay, Mrs. Abesiri, can you recommend something that what is new with your findings?
Yes, madam. I recommended these in our well water samples. In my study earlier, I detected these cyanotoxins in well water samples, drinking water samples, and therefore some samples exceeded above the guideline limits value. Therefore, it may be the reason for kidney failure.
Okay, thank you very much, Ms. Abesiri. Okay, we will move on to the next. Do you have any clarifications, the audience? Any questions? Okay, so we will move on to the second presenter. I would like to invite Ms. Disanayaka. Is she there? Yes. Ms. Sangamali?
Yes, I'm here.
Yes, so they will give us the presentation. They will share the presentation.
Good day to you all. Today I'm presenting my research. I'm Sandhamali Sanayaka. This is my research. Geospatial analysis about habitat preferences of Odonata within the DSR wetland park. This is my research topic. Next one. This is my content. First one, study area. Study area is located in urbanized area. which is the administrative capital of Sri Lanka. The park consists with marshes, lakes, ponds, water bodies, and flooded woodlands, and they have so many species of animals. I think I have to introduce the dragonflies. Dragonflies and damselflies, both of sensitive to aquatic life, and they have pictures. One is a dragonfly. They have spreading their wings. This is a damselfly. They have not spreading their wings. Next one, this is my study area. The water accumulation area was my study area because they have huge area, 64 acres. I have done only 24 acres. Method and materials. It is very important to my research because firstly I have done recording the ordinate species. So I use land transact survey because they have so many footpaths. And next one, all ordinate species observed directly and indirectly. Directly means using my eyes, and indirectly means I have used binoculars because some ordinates were far from me. Next one, they have some water bodies. I could walk in around this path and taking pictures and observe their habitat. Backside of observation are very important to me because all ordinates they do their activities around the bank side. Next one, data collecting. Data collecting methods I have done 6 to 12, 3 to 6 because some species activating in the evening, like this picture show you dingy dusker flyer activating in the evening. A total of 11 months of field work were conducted from 11th February to 20 January 2021, and I have selected 3 days randomly selected every week. Endangered binocular and the camera and GPS machine were used for the close observation, spotting the location, obtaining photographs, and use some guide for the identification the ordinator and to record their residential status. Results and discussion. I have recorded 330 ordinates. I think we can see in Sri Lanka 129 species. I have recorded within this park 330 belongs to 5 family. All identified species categorized according to their residential status. I have mentioned 2012 red list. I have used to categorize this residential status. This is the these are my recorded. These numbers include 1 endemic species, 2 endemic subspecies, and migratory species. I have mentioned endangered 1 species, vulnerable 2 species, and near threatened 9 species. Especially I have to show you. This is endemic species. I have recorded Nibbly 1, and this is migratory species and also resident. They have can see October very huge flock in above the sky. This is distribution of Ordinata in DSR wetland park. You can see the brown color, and you can see they are next to the river bank sides. You can see in this map. According to the developments, high-level distribution can be analyzed near the bank sites and canals. But some individuals distribute in other areas within the premises as they prefer the more suitable habitat. Some dragonflies used without water, like Prunus bloodtail, and some special characteristics were observed, like Malay scotter damselfly used floating leaves of aquatic plants to live, and sometimes they hanging on the some plants in the evening on the land. Mating and egg-laying habitats, I have spotted these habitats. You can see they are mating, egg-laying, and mating time, the heart position. You can see birthing habitat. You can see they are coming near the water body. This is also predating habitat. You can see different resting habitat. You can see also new species I have recorded this park. Also, I have used some analyzed method, hotspot, kernel, inverse distance weighted. They have, I can analyze from these analyzers used for taking the clustering density and predicting the location of their habitat. You can see this map, maximum diversity analysis of Odonatine research area. You can see. And conclusion, I have to tell, Odonate species are very sensitive of the water quality. These are not qualitative to their aquatic life. And we have to cutting edges from using their taking their habitat spotters area, protecting their areas. We have to protect their areas, and when we remove the aquatic plants, we have to protecting their habitat because they are digging these areas, and we can do a awareness program about the ordinate habitats to protect them. And thank you all.
Thank you very much, Sandamali. Sandamali, can you on your video? Yes, it was nice study. What you have done, this is as a special degree program for the...
I'm in Humanities and Social Science of University of Sri Javadhanwa. I have done 3-year gender degree. Main subjects are geography and statistics. But my interest in environmental, because I couldn't do the science in my A-level because my family problem. So
you have done this as an interest.
Yeah.
For your interest, no?
Yeah.
Okay, that is good, because you are doing this GIS and all that. You have shown some maps, but if you can give us the enlarged maps, that will be ideal, because it's very difficult to see the habitat utilization of the dragonflies, right? What you were telling, that these dragonflies, they used to stay near the riverbanks and they need the water bodies for their breeding purposes. So as you mentioned, and you were telling that their favorable habitats are banks. Okay. So with your data, how can you come to this conclusion? How did you conclude that? The banks?
Because I have observed they are in sometimes they are in a group. When they active in their mating and the resting, they are doing all things around the water body, and most of time they were bankside, hanging or mating.
When you are taking the data, have you obtained those data also while you are doing the line transect? Line
Yes,
transect.
not my transact. I use point and some other walking around.
So you have observed so many interested behaviors along
Yeah.
along this. And other question is, you have done this clustering of odonata and this kernel density. What are the conclusions that you could come with this?
When using kernel density and those IDW, we can use the favorable area they were in area of the park. It means distribution of coordinate habitat, most suitable habitats of there.
Okay, so and you were telling there are some other suitable habitats also. What are those?
Sometimes some ordinates located in a ground area. I think when I take in a tree like Mada, they are hanging in night.
Okay, for
Sometimes we
resting
can see.
purposes. So that means they went out of these water bodies for their other activities like resting and all, right? Okay, thank you very much. And do you have any other questions, the audience? Yes, Sandamali, thank you very much for presenting your interested data. Thank you. We will move on to the next presentation. That is Mr. De Silva. Mr. or Miss?
Present, madam.
That is, sorry?
I'm here, madam.
Okay. So they will share your presentation. Thank you.
Good day, everyone. I'm MLID Silva, and my research topic is preliminary evaluation of extent and structure of illegal plant trade via cyberspace in Sri Lanka. These are my contents. Introduction. Both illegal as well as legal wildlife, it is considered as a growing threat to global biodiversity and species conservation. So this illegal trade is powered by the demand for pets, market for luxury goods, food, ornamental purposes, and other traditional medicinal purposes. This internet has become a major mode to conduct wildlife trade, including plant varieties. So over the last years, in the area of conservation science, the plant crime or the illegal plant trade has been considered as a major environmental problem. This modern trade is being motivated or reshaped by changes such as increased access to wildlife, trade exposure, and modified infrastructure such as availability of active internet connections. So with the technological advancements, this trade has expanded massively through creation of new and hybrid marketplaces. So the internet has changed the way wildlife is traded, and where this open web has exposed the trade, but this exposure is not enough to monitor these illegal businesses that is happening via social media, and it is more challenging, but it is with a very great importance. Objectives. Since this is an emerging and important global concern, our objectives was to investigate the potential threats posed by social media platforms to biodiversity of Sri Lanka. And secondly, our objective was to investigate the characteristics of illegal plant trade happening via social media platforms in Sri Lanka. Methodology. The methodology we followed was direct observations in social media platforms and in other local advertising websites from 2019 to 2021. Here we observed major and popular social media applications in a daily basis during the study period. Then we recorded all the information such as traded species, location, price, demand, and the interest availability, thoughts of the interested people in social media platforms. Results and discussion. total of 87 internet posts during the study period were observed, including 9 plant species. As you all can see, table 1, it shows the recorded traded species and the level of protection received by Flora and Fauna Protection Ordinance to these recorded species, and it is clear that all the species are under protected category. And figure 1, it shows the national conservation status of these recorded plant species. Here you all can see majority of the species are either endangered or vulnerable as per the 2020 Red List report. And when you observe the traded plant species, it was found that all these species are belonging to 4 plant families, and majority were from the family Orchidaceae. According to figure 3, a clear correlation can be observed between the number of posts and the month of the year. For example, it was found that posts requesting or selling foxtail orchids were high during the months of November, December, and January, which marks the flowering season of this particular orchid species, which is shown by the blue arrows. And same scenario was observed for Vesak orchid as well. For example, the highest number of posts for Vesak orchids were during the months of April and May, which records which marks the flowering season of Vesak orchid. And it is shown with the green color arrows, and with the red color arrows, I emphasize Raddotalu, which recorded the highest demand during the months of December and January each year. And figure 4 and 5 shows the results obtained by interviewing traders in social media. Here it was found that 75% of the interviewed traders showed the interest to have native and endemic plants in their collection. And from that, 40% of them confirmed the availability of endemic and native varieties in their collections. This slide, it shows some posts on requests and posted with the intention of selling native and endemic varieties via social media. Here we have Nepanthes, Mahahedea, several orchid varieties such as Vanda tessellata, Vanda testacea, and Raddotul also here. Conclusions and recommendations. An emerging enthusiasm and demand was observed on illegal wildlife trade via social media platforms. Trading of native and endemic plants for ornamental as well as luxury goods were observed. Orchidaceae family plants are the highest traded species, and majority of the species were belonging to either endangered or vulnerable status according to national conservation status. High demand during special religious occasions and in flowering seasons were observed. For example, December and January, with the beginning of Sripada season, there was a high demand each year for Raddotalu. And intentional scams, misidentification, substitution of commercial varieties as native varieties were observed because native varieties, some native varieties, though they have similar commercial varieties in the market, people prefer having native varieties over these commercial varieties. So there are some scams we identified. They regarded as ornamental varieties as native varieties. So well-established organized traders as these scam people and random traders were observed. Awareness programs popularizing similar commercial plants and strict social media follow-ups are highly recommended as a tool for combating this illegal wildlife or plant trade happening via social media platforms. These are my references, and thank you so much for hearing. And comments, suggestions, and questions are welcome. Thank you so much.
Thank you very much, Mr. De Silva. And now our findings. Actually, the illegal trade is very much high in these days because of, as you told, that with this increasing our knowledge with this internet and facilities are increasing and all. But anyway, what you were telling, the fauna and flora ordinance, we have protected some plants, no? But most of these plant species that are trading are in that list, isn't it? Can you on your video, please? Yes.
Yes, madam. Most of the species are under the protected list, protected category.
Yeah. So you are pretty sure that they are still going without the permission from the wildlife department?
Yeah, what we actually followed was whenever we come across with a species or a trader, we chat with misguided profiles or fake profiles. So we request them, what is the place you collected? Is there a license or some kind of conversation is built up? So we came across to know that none of them doesn't have any permit issued by the Department of Wildlife. Actually, I'm a fisheries student. So since I'm from Ocean University. So this actual wildlife, this plant trade is a branch of wildlife trade happening there. So we are doing a huge, very comprehensive research on wildlife trade, and this plant trade is another branch in this research.
Yes. So how can we accommodate, or how can we get rid of this trade, or how can we stop the wildlife department? What can they do against this? Because we know that if we are doing any research, we take the permits and for to extract or for do the genetic study or whatever it is, we have to take the permission. But still, they are doing these things without this permission, and the wildlife department, the people also handicapped with this arena.
Actually, madam, though I haven't included that part in this abstract or in the presentation, there was a very interesting finding that we came across. The thing is, there are several wildlife pages maintained by department officials, such as DWC, Apepitua, some kind of groups and pages. And also there are groups and pages maintained by university students who are interested in ecosystems or ecology or wildlife things. So whenever the posts regarding the value, regarding the law implementation, for example, when the wildlife people, they post arresting of illegal trade and what they have done, what is the fines and what are the consequences, then we observe whenever the number of posts or the activity of the wildlife officials or the interested parties increase, number of posts of the illegal traders decrease with a very good correlation. All around the 4 years, we observe that whenever in the month of January, when the post of the wildlife is increasing, number of posts of the traders are clearly decreasing. There is a positive correlation, and we are really sure that that's the point we have to address. Whenever we do the awareness sessions or the other dissemination of knowledge is happening there, then we
have to give the message. We have to give the message to the traders. It's a good point. Yeah. And you have observed some demand changes within the months. Why do you observe that? What is it? The plants are increasing, or they are flowering seasons, or what will be the reason to see these differences?
In majority of cases, that's the flowering season, madam. And also, especially in the Rupicola, Raddo Talu, there is a great increase of demand during the beginning of Sripada season, as well as during the beginning of January, because in the
winter,
for the religious purposes. people, they seek Raddodolu, like there are beliefs there that this is a very religious plant and the Lord Buddha and the stuff there are. So people are highly demanding that species to offer to the temples, as well as Sripada, Ruwanali Mahasaya and Sri Dalada Maligav. They specify those areas in the beginning of every year, as well as in the beginning of every Sripada season. There's a
But
huge,
that means, sorry. Continue.
There's a huge demand during those months. So
that means they are trading, they do this trade within the country, not in the at the international level. What do you think?
there are some posts that
Local level also.
Yeah, there are some international levels, especially reptiles and bird varieties.
For these plants, most of the plants they are selling within the country.
Yeah, definitely most of the plants are selling within the country, madam. We didn't came across with any crime or post regarding internationally trading of plants during the study period.
Good. If you can give some, what do you think about the fauna and flora ordinance? They have given enough protection, isn't it? Do you think that we have to increase or we have to do something against that? Or?
Definitely, madam. In terms of plant as well as other wildlife, especially with the majority of the birds, there are so much loopholes.
But you know, the birds, they have given a clear-cut, unprotected list. And likewise, all the fauna and flora, we have given this wildlife, this protection ordinance has given the list. But still, you think that we need to upgrade that?
Yes, ma'am, definitely, because even, for example, that rostering parakeet is an unprotected species.
Yes.
But in the amendment, it clearly says if someone is trading the species, he or she has to have a permit.
Yeah.
But the department officials, they doesn't have that knowledge. It's really, really sad to say this, madam. I have seen traders claiming, and even the wildlife officials, we interviewed them. They claim they are non-protected and there's nothing we can do. That's really sad. There is a huge loophole there. Some officials, they know what to do. They even try to have that species to their hands, or try to arrest that man, giving some other points, low points. And some officials, it's really sad, simply they just let those traders go away.
Okay, so thank you very much, Mr. Silva. Okay, very interested. And so you have given so many informations, and we will do our best to implement those things and to give these messages to the wildlife department. Okay, thank you.
Thank you so much for the opportunity. Okay.
Next presenter is Mr. Sandaruan. Yes, so they will share your presentations. Estimation of invasive plants in Bundala National Park, Sri Lanka, using the GIS and remote sensing techniques.
Hello everyone, I am Charu Sandaruan from Modern University. My research topic is quantitative analysis of invasive Forestry julipora in Bundal National Park using GIS and remote sensing techniques. This is my overview of my research presentation. Let's know first, Forestry julipora is invasive species that shows aggressive growth pattern. Bundal National Park also became a threat of the Forestry julipora. Forestry julipora introduced to the 1915 Bundal National Park that affected the native species of the park. Still now, Forestry julipora coverage within the park is still unknown. So this study was conducted to identify the spatial and temporal distribution of the Forestry julipora in Bundal National Park using remote sensing and GIS techniques. This image shows the appearance of the Phosphosis julipora in Bungal National Park. These are the objectives in my research. Main objective was to examine the extent of the invasive Phosphosis julipora in Bungal National Park in Sri Lanka. Specific objective were to determine the spatial variation of Phosphosis julipora in Bungal National Park and to estimate the temporal variation of Phosphosis julipora invasion during the past decade at Bungal National Park. These are the park premises as well as my study area. I use primary and secondary data in my research. So GPS data was used as primary data. Google Earth image and satellite image were used as my secondary data. So satellite image obtained from USGS website from 1988 to 2021 under the certain criteria: quality of the image, availability of the image, and cloudness of the image. Image pre-processing techniques provide the atmospheric correction that provide the improvement of the accuracy of images, that reduce solar illumination and cloudness of the image as well. Geometric correction provides the image fitted into the WSGS 84 node coordinate system. Principal component analysis provides to select the best band for analysis, as well as unsupervised classification done by isocluster classification tool using for the classification. Ground truth data and Google Earth image used as a validation process, as well as in spatial distribution, we create the map of the Phosphosis julipora growth pattern and other land use changes. In the change section, we make the graph how to change the square kilometer of the Phosphosis julipora in Bungal National Park. In statistical analysis, that shows tendency of the Phosphosis julipora. In the last step, discuss how to effect what are the reason effected to the Phosphosis julipora for develop the aggressive growth pattern. These are the image from 1988 to 2016. Clearly shows the aggressive pattern of the Phosphosis julipora. Phosphosis julipora represent as a red color. First image is 1988, second 1995, third 2001, fourth 2009, fifth 2016. This time series data clearly shows coverage of the Phosphosis julipora with time to time. In 1998, 9.4 square kilometer, and 1995, 19.3 square kilometer, and 2001, 27.5 square kilometers, 2009, 35.4 square kilometers, and 2016, 32.9 square kilometers were represented. So this image clearly shows aggressive growth pattern of Phosphosis julipora in Bungal National Park. According to the availability of the data and query function that we fitted to the data, we can observe that Phosphosis coverage area had been growth with diminishing speed. As the result, from 2009 onwards, coverage has started to reduce. These are summary of my research. So, Balan and scrubland and dense forests were gradually affected by Phosphorus juliflora. 60% of land coverage of the Boondal National Park invaded by Phosphorus juliflora. So, germinated pods were released with pace. It easy to survive and growth. So, based on my data, uh, shown the diminution with reducing speed of Phosphorus juliflora, as well as Phosphorus juliflora highly aggressive to the waterlogged area. These are my recommendations. So, institutional and related agencies involvement are really important, as well as we can make the extra production like honey and like that. Biofuel production can be made from Phosphorus juliflora. Um, maybe give the access to the neighbors to the for the daily purpose with Phosphorus juliflora species. These are the limitations that I face. Several regions cannot be accessed in park premises. If we use a better resolution than 30-meter satellite image, it helps to perform the better map. So it's a time gap between the satellite image difficult to arrangement. That means it's effective to cloud coverage also. So that are the reference. These are the reference that I use. Thank you. This is in my presentation.
Thank you so much, Mr. Sandurwan. It was nice. There are nice findings with the Boondara National Park. Since you have told that it is a very important national park as a Ramsar site, that was the first Ramsar site in Sri Lanka, and it is very good site for the migratory birds. So that's why it was a Ramsar site. But anyway, that you have done this with this study on this invasive species. Have you noticed that any measures that they have taken, the wildlife department has taken to eradicate these invasive species?
Yes,
Control
madam.
this?
Yes, madam. So they gave the permission for the some private companies such as Sri Lanka and Mars companies to control this Phosphorus juliflora. The companies use these Phosphorus juliflora used as a biofuel products in their purposes. So DWC not get proper management for control this Phosphorus juliflora in invasive in Boondara National Park.
So what do you think proper management? So if they do their effort to remove these, and I have seen that, I have been there, and they have taken those plants and the inmates and those firms, they used to take it as a biofuel. So that is correct. So what else that they can do, the wildlife department, if you, with your knowledge, can you, so you have taken the ground truth data, isn't it? So at that time you have visited there.
Yeah.
Without visa, you were using the Landsat data as well as you have been there. So why, with that experience, what can you tell, or what are the advice or the management implications that can be?
So Phosphorus juliflora is as a invasive species, but this species used as make extra production like honey and medicine and such things. So at that moment, the 60% of the Boondara National Park covered by Phosphorus juliflora. So forest and scrubland were decreased because of in aggressive development of Phosphorus juliflora. So we can identify the area of the Symposium juliflora in Bhudal National Park. We can so we, these species we can use as biofuel production, very large scale. So that I think that that manner is very suitable for the control of the Phosphorus juliflora in Gundal National Park.
So you are advising them to what
Yeah.
they are doing, that they are taking them as a biofuel, that you want, you are telling we have to increase that work or that effort.
Yeah.
And how did you have analyzed the data to 1988 to 21, but in your results you have given to 2060 up to. In your at the beginning, you were telling that you have taken the data for 1988 to 2021. And again, but you have stopped analyzing from 2016. The data are deficient, that you didn't get any
actually, madam, I used Google Maps and find the different variation of the other land use changes and phosphorus juliflora in Gundam National Park, and that the things is used to identify the phosphorus juliflora from 1908 to 2001.
Have you observed any significant variation between these years, or this up to that you have given the bar chart as well as you have prepared the very nice maps? With that, can you just...
Between the 2009 seen with significant variation, but 2016, yeah, it decreased. So the thing is, the private companies, like I mentioned, Sri Lanka and mass companies, intervention for control phosphorus juliflora in Gundam National Park. That's the significant things I see.
With this. So don't you think that if you do this control, if you have taken, they have taken some control measures and those things, if you can give another plant which is suitable for that land, may protect the invasion or something like that? Or I don't know, I just asking.
Actually, according to my available information, but there are no predator species. That the thing is that the thing affected to the aggressive development of phosphorus juliflora in Gundam National Park. So, but we can so we can actually we can use the only one thing is control for phosphorus juliflora. The species actually used for the biofuel production. That's the things I think very better for
nowadays
control.
we can use it, no?
Yeah.
Okay, yes, thank you very much. That's so much with your effort towards the control of the invasive plants in these valuable sites, Ramsar site. So thank you very much, Mr. Sandaruan. We will move on to the next one. Okay. So the next presenter is Mohandira.
Yes, madam.
You will present about the current status of a rising feeder population of spotted deer in suburban lowland wet zones in Sri Lanka.
Hello, I am PMSR Mohandiram. For the 26th International Forestry and Environment Science Symposium, I am going to present my abstract on the title of Current Status of a Rise in Feral Population of Spotted Deer in suburban lowland wet zone of Sri Lanka. According to the Fauna and Flora Protection Ordinance of the country, axis deer are one of the protected species. Normally, their distribution is restricted to the dry zone. In figure 1, you can see their normal distribution in green color area. Few decades ago, some people in lowland wet zone reared axis deer as pet animals in their houses. Released and escaped deer from this captive condition has established a new feral population in lowland wet zone. Now, this feral population has attracted the media and social attention mainly because of the damages experienced in both side human and deer. These are some news articles published regarding that particular deer population in the study area. Regarding this study, I had two main objectives. First one was determination of population dynamic characters such as population size, age structure, and sex ratio. My second objective was identification of habitat utilization patterns of axis deer in this area. My study sites located in the Western Province, Colombo district. My two study areas were Homagama Pradeshiya Sabha and Kaduwela Municipal Council. Homagama Pradeshiya Sabha consists with 91 Grama Niladhari divisions and Kaduwela Municipal Council consists with 57 Grama Niladhari divisions. In methodology, I conducted block counts and line transects for the determination of population dynamic characters. And I conducted block counts, line transects, and spotlight counts to study their habitat utilization pattern. I conducted my daytime observations from 6:30 am to 6 pm and nighttime observations from 7 pm to 1:30 am. According to daytime census, I could record 393 total deer population from this area. Out of them, 46 were adult males, 263 were adult females, and 84 were juveniles. So, 67% of the population was represented by adult female, 21% represented juveniles, and 12% represented by adult males. According to sex ratio, there were 5.71 adult female per 1 adult male. According to age structure, there were 0.27 juvenile per 1 adult. According to my daytime census, there were 231 axis deer in Homagama Pradeshiya Sabha and 162 axis deer in Kaduwela Municipal Council. In the study area, there were 5 main habitats. They were shrubland, grassland, home garden, paddy field, and rubber plantations. Out of them, their most preferred habitat was shrubland during both day and night time. Their second preferred habitat was grassland. When considered about home garden, there is a trend of attracting home garden during night time than daytime. When considered about paddy field, I could not observe any axis deer from paddy field during night time. I conducted my night observations in March. So at that time, farmers have harvested their paddy from their paddy field. So it can be the reason for zero count in paddy field. During daytime, there is a significant difference in habitat utilization of shrublands when it compared with all other habitats. During night time, there is a significant difference in habitat utilization of shrublands when it compared with rubber plantation and paddy fields. In figure 6, you can see a pair of axis deer utilizing home garden during night time. In figure 7, you can see a group of axis deer utilizing paddy field during daytime. Their second preferred habitat was grassland. When considered about home garden, there is a trend of attracting home garden during night time than daytime. When considered about paddy field, I could not observe any axis deer from paddy field during night time. I conducted my night observations in March. So at that time, farmers have harvested their paddy from their paddy field. So it can be the reason for zero count in paddy field. During daytime, there is a significant difference in habitat utilization of shrublands when it compared with all other habitats. During night time, there is a significant difference in habitat utilization of shrublands when it compared with rubber plantation and paddy fields. In figure 6, you can see a pair of axis deer utilizing home garden during night time. In figure 7, you can see a group of axis deer utilizing paddy field during daytime. During night time, there is a significant difference in habitat utilization of shrublands when it compared with rubber plantation and paddy fields. In figure 6, you can see a pair of axis deer utilizing home garden during night time. In figure 7, you can see a group of axis deer utilizing paddy field during daytime.
Really sorry for the inconvenience. There's a small connection error. Please hold on.
In home garden during night time. In figure 7, you can see a group of axis deer utilizing paddy field during daytime. In conclusion, there were 393 total axis deer remaining in this study area. During both day and night time, their most preferred habitat was shrubland and their second preferred habitat was grassland. Their age structure indicated that there is a high growth rate of the population because juveniles represented a considerable proportion of the total deer population. Their sex ratio indicated that females are the majority of the population. So, their sex ratio is skewed towards the females. With their polygynous mating behavior, one adult male can mate with few other adult females. So the population size can be further increased. As there is lack of natural predators in the suburban lowland wet zone and the abundance of the food resources throughout the year, their population size can be increased rapidly, and it can create some severe socioeconomic problems in the future. Therefore, it's timely needed to take some mitigatory measures to control this conflict. For that, we can increase the carrying capacity of the area for axis deer, and at the same time, we can utilize some population control methods to reduce their population growth. In recommendations, daytime census is the most suitable method to monitor deer population in these areas than nighttime census. As the shrublands are the most preferred habitat, they live abundantly in those places during the day and night. To do the night census, we have to use spotlight count method. As the shrubs and trees cover the light beam, we cannot do it correctly during night time. If we can utilize a suitable marking method or radio tracking method, we will be able to find out their exact local movement pattern in these areas. To increase the carrying capacity, we can set in aside some land dedicated for them. If we are going to use translocation as a population control method, it should be done only upon collecting more baseline data. Otherwise, if we don't know their population dynamic characters, this method will be useless. Since this feral population is there for several decades in wet zone, genetically they might have taken some changes in their gene codes. So if we can compare them with their dry zone counterpart, it will be really interesting. These are the key references which I used for my study. Thank you for paying your attention on my abstract presentation. Thank you.
Yes, Mr. Mohandiram, thank you very much for your presentation. You have given a very informative informations are so much and available in your presentation with these feral populations or the spotted deer population. What I want to just ask to clarify, you have done the night sampling only 1 month. And have you compared with the daytime work you have done in much more, that means from October 2020 to 2021. Can you compare these two and come to a conclusion with the habitat use of these two, this species?
Yes, madam. Actually, the reason I conduct 3 days in March for night spotlight count. The actual reason is that I have to travel from K-Gold to Hormagam area with a cab. So the funding is I have to reduce the
that
so,
is something. There are some difficulties to do these things.
Yes,
but if you have taken or you have analyzed the data,
yes,
but you have taken only for March, day and night, so that will be okay. Don't you think?
Yes,
so?
Yeah.
So other thing that you were telling, that you have observed some skewed sex ratio differences with these polygamous species, their breeding behavior or the mating systems, mating behavior, no? So they have some differences with that. So what is your opinion with that? So there are more females.
Yes, in previous study, some in foreign countries, they have found that when there is plenty of food available, they tend to make more female juveniles. So so in this area, I think they have plenty of food, enough food resource very available. So I think it can be a reason for this skewed sex ratio. And at the same time, there is lack of natural predators. So female deer are small in size compared with male. And when considered about if there is a natural predator, there is a high. The female are most susceptible to predator pressure, but there is no predator considerable predator in this area. So female also can.
All the environmental conditions are favorable for their population growth, isn't it?
Yes, madam.
They have no predators, plenty of food, and they have the skewed sex ratio and everything. So what is your opinion to control this? We have to control it, no? So
Exactly,
that
madam.
is an invasive and the level, and they are coming to these croplands, and damage is
Yes,
very much. So what is your opinion,
to control?
Yes,
Yes,
you have given a good opinion. So what will be the best
yes,
that
madam.
we can do?
Yeah, we can suggest short-term, mid-term, and long-term solutions. When consider short-term, we can use practically we can use translocation. So we can capture them and translocate them. Other than that, in Sri Lanka, we have not used injection. We have the hormones,
Sterilize?
kind of sterilization, but it's permanent, madam. We have to boost that vaccine after 2-3 years in natural environment. So it's a kind of not suitable to Sri Lanka.
Why is it expensive? Why
Yes,
you are telling that
I think
it's not suitable?
Yes,
Okay.
So translocation is very important. Addition to that, we can if we can provide fences. If resident can make fences around their crops or their home garden very well, I think they can avoid the attack from the damage caused from coccidia. But some people, they have no enough money. Yes, madam. Yes, madam.
So what do you think about the predators, dogs and all? Don't you think that they affect on
Actually,
the
madam,
population? No.
yeah, they afraid to dog. But one night I could observe they came to home. The dogs are also close to them, but they don't attack. But sometimes, madam, juvenile can be attacked, but I could not observe any like that. But people believe that they are afraid to dog.
Okay, so thank you very much. It's a very good presentation. Okay, thank you, Mr. Mohandira.
You're welcome, madam.
So we can finish our wind up the session now. So thank you all the presenters. You all have given very good informative and presentations, and everything is good for the conservation. So the biodiversity conservation. So we will take the message up to the department level with our maximum effort. So thank you very much, all the presenters, and we will meet you again. Right. Devin?
Thank you very much, madam. Madam, if you could announce the best presenter. I think we have sent you an evaluation form.
Now, all are best. So you have to. Anyway, so I think very difficult task with all these informations and all I have given the marks. So Mr. Diram is the best presenter. Can I? Okay.
Thank you.
Thank you very much, madam.
It is very difficult, but all the others also nice. But with this statistical analysis and all that he has given, therefore I have to take that as first. So don't misunderstand and don't give up your ideas or whatever. So I congratulate all of you, including.
Thank you very much, madam. And congratulations to the best presenter. And if you could, everyone who was in the second session, if you could switch on your videos and give us a thumbs up, we would like to take a photo. if everyone could turn on their
On the
videos
video.
and give us a thumbs up. Okay, thank you very much.
Thank you.
With that, we are reaching the end of the part 2 of this interesting session on biodiversity conservation and management. And we will be starting the third session in another 5 minutes. Please, you have to rejoin again to the same link. Thank you.